Okay, so we're back with part two. I am using a different software this time. We'll see how well it works. I'm not happy with the quality of this webcam. Come on. But you work with what you got, right? Okay, so another way that we can use DNA is to determine how closely related organisms are to one another. And the order or sequence of DNA bases is different for each species. So by comparing them, we can determine how closely related they are to one another. The more similar the sequences are, the more likely it is that two organisms have a recent common ancestor. So for example, for a long time, people didn't know whether red pandas were more closely related to raccoons or to giant pandas. And when scientists compared the DNA sequences of these mammals to one another, they saw that the DNA for the red panda was more similar to that of the raccoon than to that of the giant panda. So they determined that the raccoon and the red panda had a more recent common ancestor, which you can see right here, than did the raccoon and the giant panda. A DNA sequence alignment usually looks like this. These people are comparing the DNA sequences of the different species on the left, you see here, and you can see that the bases have been aligned, so you can see where the differences and the similarities are. And it will lead to something like this. So humans, we share approximately 99.9% of our DNA with one another. So that means that all the differences we see are accounted for by only 0.1% of our DNA. We share approximately 98.7% of our DNA with chimpanzees and bonobos. We share only approximately 92% with mice. So it's pretty clear by looking at the organisms and by comparing their DNA that we're more closely related to chimpanzees than we are to mice. Excuse me a moment. I think they want food, but really they just want attention. This is Loki. Hi. It's like, your shit. Okay, moving on. All right. Stop here and do part one of the handout, DNA structure and replication. And it's not just busy work, it's a critical thinking handout, and it's going to help you with reinforcing the concepts and connecting concepts and remembering what's going on. Help prepare you for the quiz at the end. Right. By the 1950s, scientists knew that DNA existed. They knew that it carried genetic information, but they had no idea what it looked like or how it worked. And there's somewhat of a little bit of scandalous story behind this. In 1953, the structure was discovered by James Watson and Francis Crick, these dudes over here, and Rosalind Franklin, this lovely lady scientist. She was also working with another guy called Maurice Wilkins. She did most of the work, so that's why she's credited with the structure, the discovery of the structure of DNA. But let's see. Watson and Crick, they were working together in this picture over here. And Rosalind, Franklin, and Maurice were working together. And each team had different data. And if they had just put their data together, they would have found the structure of DNA right away. But Maurice and Rosalind had kind of a rocky relationship. This was a time when women were not scientists. And when Rosalind began working with Maurice, he immediately assumed that she was his assistant and began treating her that way. So their relationship was always a little strained. And then without Rosalind's knowledge, Maurice took one of her X-ray pictures of DNA, this one right here, and he secretly showed it to Watson and Crick. And as soon as they saw it, they immediately knew what the structure was, and they published a paper about it. And Rosalind Franklin figured it out on her own and published a paper too. But at that point, hers was treated as an afterthought because Watson and Crick had already dropped the bomb on the scientific community. Watson and Crick, they won the Nobel Prize for their work. But Rosalind Franklin was dead by that time from cancer, probably caused by the x-rays that she had worked with to find this structure. And she wasn't recognized because the Nobel Prize can't be awarded to dead people. So that's kind of a sad story, but as time has gone on, people have realized that she was responsible for a lot of the cool stuff as well. Remember, types of biological molecules include carbohydrates, lipids, proteins, and nucleic acids. There's DNA and RNA. Nucleic acids, therefore, are a type of biological molecule. We're going to talk about DNA first. And DNA structure, I want you to go ahead and watch this animation and come back. Stop. How do I pause this? Come on. Oh my God. There we go. Okay. So let's talk about the monomer of DNA. It's called a nucleotide, and there are three components to a nucleotide. The first one is the sugar molecule, and that's right here. The second is a phosphate group, and that's this one. And then there's a nitrogenous base, and sometimes it's referred to as a base. Called nitrogenous base because there's nitrogen in it, as you can see here and here. Each nucleotide has three parts. Yep, there we go. And there's four different bases. Thymine, which is abbreviated with a T, cytosine, which is abbreviated with a C, adenine, A, and guanine, G. Nucleic acids with two carbon rings are called purines. They have two. And with one carbon ring, they're called pyrimidines. And you can remember this with the acronym CURE as gold, purine, adenine, guanine, and PICUT, pyrimidines, cytosine, thymine, and the U is for uracil, which is only an RNA. And we'll talk about that in a minute. Very important to realize that the DNA, the cell stores genetic information in these nitrogenous bases. So when you see a string of letters like this, for example, in DNA alignment, these letters represent the bases and the order of the bases. And if we know the order of the bases, we can actually tell which amino acids the DNA is calling for and therefore which protein it's going to make. Each three amino acids is called a codon because it codes, each three nitrogenous bases is called a codon because it codes for specific DNA, a specific amino acid. And TCA, that calls for one amino acid. AAG, another amino acid. ATT, another amino acid, and so on and so forth. If you want to, you can go to this website and take a look at this gene. It's incredibly long, and it's just super cool to be able to go to a website and look at all the genes that scientists have decoded, which is cool. So the DNA bases are different shapes, and that is how the cell tells the difference between the bases, also by the location of the hydrogen bonds between them. So when it's accessing the DNA, it knows which base it's looking at, and then it knows which amino acid is going to come next in the protein that it's making. The order of the DNA bases determines the order of the amino acids. The shape of DNA is called a double helix because there's two backbones of sorts, and they twist around each other in a helix shape, which is a spiral staircase shape. It looks like a spiral staircase because the bases are in the middle, like the steps, being protected from the environment, and the backbones are on the outside, sort of like the handrails. And if we unwind this double helix, it forms a ladder shape. Here's our nitrogenous bases forming the steps, and they're held together with hydrogen bonds. Notice that thymine always pairs with adenine, and guanine always pairs with cytosine. These are called the base pairing rules. It's G with C, A with T, and you can remember that by the catchy acronym GCAT. So sometimes people will talk about the base pairs. What that means is just a pair of opposite bases. For example, A to T or G to C. Those are base pairs. And when we refer to the length of a DNA molecule by the number of bases, we could say base pairs or we could say bases. A gene for an SRY protein, which is the sex-determining region Y protein, that's 92,657 bases, or we could say base pairs, long. And remember, what's very important about this is that the cell can store genetic information in these nitrogenous bases. And when it reads the genetic information, every 3 bases code for 1 amino acid. You could think of each base as a letter and the order of bases as a sentence that means something, because it's the order of the bases that tell the cell which amino acids should be used and therefore which proteins should be made. Now pause the video and use the base pairing rules to fill in the other side of the DNA molecule. And what you should get, pause. Okay, what you get over here is G. You should have A, you should have C, you should have T, you should have G, and you should have T, because those are the base pairs. I want you to note that the sugar molecules and the phosphate molecules right here, these guys bond together to form the outside of the DNA. We call this the sugar phosphate backbone. The sugar phosphate backbone is held together with covalent bonds between the nucleotides. Those are very strong. And the nitrogen bonds in the middle of the molecule here, hold the bases together. Those are weak. And this is important because when it's time for the cell to access the genetic code, it's going to have to open up the DNA molecule, and it can very easily unzip the DNA molecule on the inside by breaking these weak hydrogen bonds. But the DNA sides, either side, will hold together because of the really strong covalent bonds on the sugar phosphate backbone. Each one of these units is a nucleotide. So we've got a nucleotide here. See, there's the phosphate group, the sugar, and the base. We've got another phosphate here, and here, and here. So just notice that the nucleotides on one side of the molecule are facing one direction with the phosphates pointed down. And on the other side of the molecule, they're facing the opposite direction with the phosphates pointed up. This becomes relevant when we're talking about the direction in which the DNA information is read and copied. And it has to do with the sugar molecule. My mouse, right here. You might remember from chapter 3 that at each corner of a molecule, we assume there's a carbon atom. And the carbon atoms are numbered clockwise like this. So this is the first one. We call that the 1'. Second one, the 2', the 3', the 4', and then the 5', because this is the last carbon atom of the sugar molecule. Notice that the H, or it's going here, it should be an OH. There we go. I mistyped that. OH, it comes off of the 3' end. Like you can see it here, it's coming off 3' carbon. And also notice that this phosphate group is coming off of the 5' carbon. This gives each side of the DNA molecule a direction. So on the left side over here, you can see that the 3' carbon is the last carbon of the molecule right there. That's the 3' end of that strand of DNA. And if we follow it up, you see that it stops with the 5' carbon. So we call this the 5' end of the molecule. And on the opposite side, it's the exact opposite direction. It's the 5' end on the bottom and the 3' end on the top. Because of these opposite orientations and the fact that the DNA, the opposite orientations and the fact that the DNA flows in opposite directions, we call DNA as being anti-parallel. Go ahead and watch this animation and then come back when you are done. So why would we even care? It's important in DNA replication when we're going to talk about in the next section. The protein, the enzyme that puts nucleotides together to make new DNA strands, can only add nucleotides to the 3' end of an existing molecule because it needs that OH, it needs that hydroxyl group, the protein, the enzyme, it needs that to grab onto. So the new DNA chain will grow in a 3' to 5' direction because we're starting with the 3' and we're ending with the 5'. So it's going to grow in this direction. Starting with the 3' and replication is going to proceed in that direction. And then on the other side, we're going to start with the 3' and go that direction because the enzyme has to have that OH to grab onto. Here's how to find the 3' and the 5' ends. On one strand, you find a 5' by looking for the phosphate group, and then on the exact opposite side, there is going to be a 3' end right there. Sometimes we write DNA like this, where we're just writing 2 simple strands, and you can always just choose and then label the 3' and the 5' ends by writing them opposite of one another. So let's just choose this to be the 3' end of the top strand. So that means the bottom strand exactly opposite will be 5'. And on the other side, because the 3' is over on the left here, this is going to be 5'. And then exactly opposite of that, please excuse my cat, on the exact opposite that is 3'. All right, now we're going to talk about structural differences of DNA versus RNA. Excuse me. Wait, pause record. All right, structural differences between DNA and RNA. First is the nitrogenous base. Oh, wait, first, as a quick note, viruses, most of them don't have any DNA. They only have RNA inside of them. And the COVID-19 virus is one of these. It has a pretty big genome for a virus. It's 29,890 base pairs long. Anyway, DNA and RNA have 3 main structural differences. The first is that in RNA, there's uracil, which is a different base, and it replaces thymine in RNA. So wherever you would see thymine, you see instead uracil. The second difference is that the sugar is different. In DNA nucleotides, the sugar is deoxyribose, right there with no oxygen. And in RNA, the sugar is ribose. The difference is right here on the 3'. So in ribose, we have oxygen, hydrogen, and in DNA, we just have a hydrogen, deoxy, because deoxygenated. And the third difference is that a DNA molecule is a double helix, double-stranded, and an RNA molecule only has one side. It's a single strand. This is because every RNA molecule is just a copy of one side of the DNA. And the fact that RNA is single-stranded, that allows it to fold up into different 3-dimensional shapes by its own nitrogenous bases being attracted to each other by hydrogen bonds. And this 3-dimensional RNA molecule shape can do various jobs around the cell. Some of them even act as enzymes. And that means they help speed up chemical reactions. This is Khaleesi with her nucleic acid hair. She has RNA over here and DNA over here. All right, DNA replication. Why? It's important because anytime your body makes a new cell, it has to have the exact same genetic information as the parent cell. And just after a cell has divided, it's going to grow to mature size in the G1 phase. And after that, in order to prepare for dividing again, it's got to double its DNA. This is the S phase. Okay, after that, there's the G2 phase, final replication for division, then division, and back to the G1 phase. The S phase is what we're going to focus on for the rest of the day. This is where all the events of DNA replication happen. Watch this animation first. It says it has experts rewriting science textbooks. It doesn't really. I don't know why they say that, but what they do is they put fluorescent tags on individual nucleotides, and then they can watch as the nucleotides are put together into DNA molecules. It's a really cool animation. DNA replication happens in the nucleus, which would be here in a eukaryotic cell, or in the nucleoid area, which would be here in the prokaryotic cell. Hey, don't eat that. Stop. I love them, but so basically it just happens wherever the DNA is found. You can see here that DNA replication is called semi-conservative. That means half of each original molecule is preserved in the new molecule. You can see this is the original molecule, and the new strands are shown in purple. So each original strand is used as a template. Watch this animation and come back when you're done. It's important for DNA replication to be able to happen in multiple places along the same DNA at the same time, because there's so much DNA, it would take forever to copy if only one enzyme were doing the job. In human cells, remember, there's 6 feet of DNA, and it would just take forever. It would take way too long if there was only one group of enzymes doing the job. Watch this animation and come back when you're done. Ooh, this is a very important one. This animation will help you visualize and remember the direction in which DNA replication happens. And I've included a little screenshot from the video to use as a visual when you study using this PowerPoint. The thing is that this guy has the luxury of a whiteboard, whereas I do not. So go watch him write on his whiteboard and explain it, because he does a really great job. It's only like 3 minutes long. So the enzyme that makes the DNA is going to grab on to a 3' hydroxyl group, OH, and then go in this direction, gluing together new nucleotides, which we say DNA is written from 3' to 5'. And at the same time, DNA replication could be happening on the other strand in the other direction. In fact, it probably is, because anywhere an enzyme can grab onto a free hydroxyl group, it can just zip right on down and make the DNA. All right, here we're actually talking about the process of DNA replication. Very important. I am not going to tell you what the steps are, and I'm not going to tell you what these things do. Instead, you're going to have to watch a demonstration and an animation. At the end, you should be able to answer these questions. And if you can't answer them, go back and watch it again. All the answers are in there. So first, you're going to watch the demonstration, and I will put a link for that. So go watch that and come back. And now watch this animation, and you can answer those questions. Hint, hint, some of them might be on the quiz. If you want to, I know this says high school science, DNA replication, but it's a pretty cool little online activity, like a click and drag kind of thing, where you get to see what DNA replication, how it happens, and it reinforces the base pairing rules as well. Ooh, this is what it actually looks like in the cell. This is very cool, made by some people at Harvard. Remember, enzymes that make DNA grab on to the 3' end and continue to the 5' end, and the same on the other side. Right, now complete parts 2 and 3 of the DNA structure and replication handout, and then take the quiz, and you're done. Stop. Okay.